Hello, iGEMers. What a fun day yesterday was, wasn't it? I was so honestly impressed of the passion and the excitement I saw in every single one of you that I was able to speak with yesterday. I'm really excited to share this talk today because this is something that's personally incredibly passionate to me. And this was being able to be a part of the first personalized CRISPR therapy that was used to treat a patient this year. So I'm going to tell you the story of IDT specifically being a part of that. And this has honestly been probably the most rewarding part of my 20-year career in this industry. So to start, let me give you a little bit of background about who IDT is. So IDT was acquired by Danaher Corporation in 2018. And Danaher is a company that has three distinct platforms: our biotechnology platform, our life sciences platform, which is what IDT is a part of, and then our diagnostics platform. And I really like this quote that our CEO, Rainer Blair, has said up here, that the Danaher you see today is purpose-built to help customers solve some of the most important health challenges impacting patients around the world. And that's really what we were able to do this last year. So IDT specifically has been around for almost 40 years. And you may know IDT as where you get your primers, but really IDT covers a broad breadth of applications. We have a lot of innovation in products in synthetic biology, in CRISPR gene editing, which is what I'll focus on today, in next generation sequencing, and in PCR. IDT is based, our headquarters is in Iowa, which is in the Midwest, but we have production facilities all across the world, and we have R&D facilities all across the US. So I actually started as a research scientist at IDT back in 2006, and I now lead our applications development team, and we support applications across gene editing and gene writing. And this is a slide that has just a lot of fun stats about IDT, but I really wanted to point out the stat up in the top right that IDT does a ton of innovation. We have scientists, about over 300 scientists have their PhDs at IDT. Many, many others have their masters and their bachelors, so a lot of higher level education and large R&D and development teams doing innovation to help the broader scientific community. And we have over 200 patents that have been granted. And another fun stat in the bottom left is IDT is an impressive manufacturing powerhouse. We synthesize over 130,000 oligos every single day, which is really, really incredible. Okay, so I really want to focus today on our CRISPR innovation journey. So as I said, IDT was founded in 1987 as an oligosynthesis company. And like I said, I started in 2006 as a research scientist studying gene knockdown, so using RNA interference and antisense oligos. And I distinctly remember in 2012 when our CSO, Dr. Mark Bilkey, brought me into his office right after the first CRISPR publication was published. And he said, "This is huge. This is going to change the world, and we have got to figure this out. We make RNA oligos. We have really got to figure this out. CRISPR requires an RNA molecule, and this is going to be groundbreaking." And I reflect on that a lot because he was absolutely right. And so really from that day on in 2012, we have had a large team really focusing on developing CRISPR components that make it much easier for all of the brilliant scientists and clinicians in the world to be able to do research. And I'll highlight a few of those things on here. So actually, I should know, I actually was the first person at IDT to do a CRISPR experiment. And I think back, especially being here today, I did that with one of our synthetic biology reagents, a G-block, a double-stranded DNA fragment that was running off of a U6 promoter that then, was translated in the cell, because at that time, making long RNA oligos wasn't efficient. And so since then, we have really optimized our ability to synthesize RNA oligos. So now we can make a guide RNA up to 150 bases in length in under 3 days. And so you can get that incredibly quickly, high quality synthesized guides. And it's kind of highlighted across this timeline the various times we've introduced new guides. We also have a large protein engineering group at IDT that focuses on helping evolve new proteins. And so in 2016, we provided our wild-type Cas9 nuclease, which was a very efficient way to deliver the guide RNA and the Cas nuclease as a ribonuclear protein complex. But then we evolved on that. We did bacterial mutagenesis, and we figured out how can we make this Cas9 more specific. And we launched our high-fidelity Cas9, which is actually now available as a GMP guide or nuclease that can be used as a therapeutic. And so that's known as our SPI-Fi in collaboration with Aldevron. And just kind of touching on a few other things here, IDT has done a lot of innovation in the HDR space. We've developed new tools. And I want to, in 2021, where it says the RAMP-seq CRISPR analysis system, that's what I'm going to focus a lot on today. As we've developed our new reagents or new tools in the CRISPR space, safety has always been incredibly important to IDT. So anytime we've developed a new protein, we've always assessed the safety profile. And to do that, we've really optimized and refined a lot of the off-target assays. So when you're delivering your CRISPR components, you want it to, the guide RNA, it's targeting where you want to make that edit. But what else is happening? Are you making edits anywhere else? That's something which are known as off-target edits. That's something we've really studied and figured out the best way to assess, and I'll discuss that today. And then I'm really excited to highlight in 2023. Now, CRISPR, as my CSO said, this is going to take the world by storm, and it really has. It is. You heard, of the launch of the Casgevy therapy for sickle cell, and it is being moved into a therapeutic in many clinical programs. So IDT in 2023 launched our therapeutic oligo facility. So we're not just making quick research products. Now we're able to make very high quality GMP grade guides. And so as part of that, in 2025, I highlight in the second bullet that says off-target analysis service. It's kind of back to those off-targets. Now that we're supporting making these GMP grade guide RNAs, we've had our customers, our clients come to us and say, I see all the great off-target work you guys have done. Can you help us? Could you do this for us? Like this is a lot of work, and it's something we need to put together in our investigational new drug packages. So earlier this year, I led the team that was able to take all of these off-target assays that we've developed and worked on at IDT, and we're offering them as a service now. So our customers can do their CRISPR edit, send their edited cells to IDT, and we can actually run these assays and get you a report of where this is editing in the genome. And so I bring this all up and kind of give you this background because then in the top right, in 2025, we were a part of the world's first mRNA-based CRISPR therapy, and IDT did a lot of the off-target work with that. So that's what I want to really focus on today, because this was true innovation for a novel nuclease that IDT was a part of. So we've been thinking about, how do we treat rare diseases using CRISPR for some time now. And in January of 2024, so almost 2 years ago, we announced this beacon for gene-edited cell therapy where Danaher, so specifically IDT and Aldevron and Cytiva and other Danaher companies have partnered with the Innovative Genomics Institute run by Jennifer Doudna and Fyodor Urnov to really figure out how to platform and be able to use CRISPR as a cure for these rare diseases. And so the goal here is over 4 years to try to develop gene therapies for 2 rare genetic diseases and really learn and build a playbook for how others can do this and how we can really be able to treat the N of 1 rare diseases. So when the opportunity came to be able to help a young boy that was born in late September 2024 and develop the world's first personalized CRISPR therapy, these teams were ready. We were motivated. We were energized, like so many of you today. Like, we have been studying this. Let's put this to work and really save lives. And so Aldevra and IDT and Acuitas, I'll get into more detail of what each of these partners did, worked together with the IGI, with UPenn, with CHOP to develop the first CRISPR personalized therapy. And this was used to treat a very rare and severe form of urea cycle disorder. And so this is a rare inherited metabolic condition that is caused by a deficiency in an enzyme that's responsible for converting nitrogen into urea. And so what happens then is this nitrogen accumulates as ammonia in the bloodstream that leads to neurotoxicity and brain damage. And this is often detected early onset of life and is prevalent in 100 children across the US. So today, current treatments are limited to symptom management and invasive procedures, so things like hemodialysis to reduce the ammonia levels, IV nitrogen scavengers, protein-restricted diets, and ultimately liver transplants. And what if we could cure this with genomic medicine? So gene therapy, as I've said, and mRNA-based approaches are evolving, and CRISPR can really be used as a potential cure to correct that targeted mutation in this enzyme. So in August of 2024, a young boy was born with a very rare and severe form of this urea cycle disorder, and specifically, it was a deficiency in the CPS1 protein. So we came together through this IGI beacon and said, can we develop this? Can we make these drug substances in 6 months to reduce risk of cognitive development and growth development and be able to actually treat this patient in February 2025? So this has really taken just the manufacturing piece, a year and a half plus down to 6 months. So how are we going to do this? So this was an incredible relationship, collaboration, teamwork between really 4 different players on the manufacturing side. So as I said, the IGI, Aldevra, and IDT, Acuitas, working with the physicians, working with the researchers at the University of Pennsylvania, working with the FDA. And so what are the challenges on the manufacturing piece we had to overcome? We need to make a new guide RNA that is specific to this patient with a specific modification pattern that makes this work well. We need to do this safely with all the correct tests in place. We need to synthesize a novel base editor. This is, I'll get into base editing here in a minute, but this was a novel base editor. We need to design a lipid nanoparticle formulation that will be able to encapsulate these 2 components and deliver this to the liver. And then we need to do, as I mentioned, we need to do comprehensive off-target analysis. We need to make sure we are not editing anywhere else other than this particular mutation in this patient. And then we need to align everyone together. Everything's got to be ready at the right time. And this, again, all in 6 months. We need to work very, very closely with regulatory and quality and the FDA to also have that ready, that we're getting the right quality plans in place, the right risk-benefit approach here. And then ultimately do this in 3 times faster than standard. So what exactly did we manufacture for this drug substance? This kind of highlights the typical timeline up top of what that takes. But as I said, Aldevron, also a part of Danaher, Aldevron made the mRNA for this ABE8E base editor that the UPenn team had been working with. So at the same time, IDT is making the GMP guide RNA. So we're making the GMP mRNA, we're making the GMP guide RNA, we're working with the FDA, we're working with the scientists to ultimately get the drug substance built that then needs to be encapsulated by the Acuitas a lipid nanoparticle. And I'll note that this particular lipid nanoparticle had already been validated in a clinical program, so this allowed us to de-risk that portion of it and be able to use something that had already gone through a program. And then ultimately then have that encapsulated and have this as a drug substance that could be the therapy for this child. So what else was going on as this was happening? Pretty immediately after birth, the amazing scientists and clinicians diagnosed this child with the particular mutation in the CPS1 protein. And right after that, the scientists, the researchers at UPenn were basically on call. They had been studying this disease, this variant. They had been studying how to treat this, and they were ready. So they took the patient-specific mutation and actually developed a cell line right away to start working on this so that they could optimize the right CRISPR editor to use, the right guide RNA to use, and then quickly after that developed a patient-specific mouse model to have an in vivo approach to study this. And then, as I said, IDT and others started doing the off-target work. We have the guide RNA they're using that is specific for this patient. Okay, we have to nominate where is there potential that this is where else this is going to cut. We started having meetings with the FDA. So partners from every one of the companies we talked about were helping support these meetings, these Interact meetings, getting the quality plan in place. And then in month 5, the first toxicology batch of this therapy known as K-ABE was completed, and we were able to do non-human primate studies. So this was really escalated and allowed to be done quicker, again, by the de-risking of the acuitous LNP. Then continuing to move on, we had the clinical batch ready, as I said, in 6 months. So it was ready. We were able to test the clinical batch in cells and then do off-target editing, off-target conformation, which I'll get into here in a minute, on those actual clinical batches and make sure any of those sites we nominated are not editing anywhere else. Continue working with the FDA and then ultimately submitting our IND in month 7 from birth, being approved and able to do the first dose of the patient, 7 months of life, and then a second dose a few weeks after that. So all of the partners that were involved in this are listed on the bottom, but what I want to highlight is this was an amazing collaborative effort between academia, industry, the FDA, et cetera, to really get this all together in the timeline safely and to be able to help this child. So the last part of my talk, I'm going to focus on the piece of the off-target analysis because that's the piece I was fortunate enough to lead the team at IDT that did this work. And so we were able to condense what typically takes over 12 weeks to do off-target nomination and confirmation in under 6 weeks. And as I said, we'd been working on these assays also. We were ready. We were super excited to put this service we have just built to use for an actual patient. And let me show you a little bit of details about how the off-target nomination confirmation services work. So there are multiple platforms that have been used for gene editing therapeutics. It started with zinc finger nucleases that have like the genomic specific region in the protein, not in the guide RNA. Talon's meganucleases, and then as I said, CRISPR really was discovered in 2012. And since then, scientists, innovators have continued to evolve on CRISPR. We now have what we refer to as our next generation editors, so base editors that are allowed to create, an A to an AT to a CG conversion, or vice versa for a cytosine base editor, prime editor. So these are all continuing to evolve, so we have to study safety of these as well and have assays to do that. And so specifically, for this patient, the novel base editor that was used was an adenine, a form of an adenine base editor, which converts an AT to a GC. And so on the right, this is a figure from the New England Journal of Medicine manuscript that highlights this whole therapy that was developed. It shows that there's a single base variant that causes the CPS1 deficiency of an A that we needed to convert to a G. And so the base editor uses a deaminase enzyme that's fused to a Cas9 variant that then is directly able to convert a single base to that corrected base. And this doesn't create a double-stranded break, so it allows the reduced risk of large deletions or chromosomal rearrangements to happen. And so ultimately then, these adenine base editors could correct that pathogenic point mutation that was present in this child. Okay, so off-targets. It is important and it is challenging to look for off-targets. There's no gold standard of how to do this today. Whole genome sequencing is expensive, and it's not sufficiently sensitive at the levels that we really need to look to make sure there's no off-target editing happening. There's a lot of techniques that exist, but really the analytical bounds and the accessibility to them are not well known. So how do you do off-target assessment? This is really thought about as a two-step process. So first, you need to do what's referred to as off-target nomination. So you need to essentially shine a light and predict and actually nominate where is there potential for this guide RNA and this enzyme to also cut in the genome. And this really needs to be sensitive. It's critical that you're not missing anything. And then you follow up with what we refer to as off-target confirmation. And so now you are taking all of those nominated sites and you're doing deep sequencing at each of those genomic loci that you have said are at risk. And this is typically done on the clinically relevant cells. And this is then allows you to assess for indels, but also any larger scale aberrations that could happen. And it's important to note that not all off-targets that are nominated are actually bona fide off-target confirmation sites. And so again, the importance of nominating all of the sites that you then look at in confirmation is incredibly important. And the field is really flooded with ways to do this. And I'll actually start on the right side. There's 3 different ways we think about doing this. And really what's important is orthogonality, using multiple of these approaches to really make sure you're capturing all of those potential sites. So you can use in silico predictive tools, algorithms that have been built based on the sequence homology of where else could this cut. You can use in vitro assays, and some of the published assays out there, we've got in boxes at the bottom. And then you can use cell-based assays. And IDT has developed the UncoverSeq assay, and I'll highlight that here. But the cellulo-based assays are really great because they have higher specificity, and they're the most concordant with off-target confirmation, and allows you to do this in a biologically relevant system as well. So how does UncoverSeq work? So UncoverSeq, as I said, is a cell-based assay, and this is an add-on to the published GuideSeq technology, where you introduce a double-stranded tag along with your CRISPR components. So along with your editor of interest and your guide RNA, you're putting this short double-stranded oligo. And so what happens is this is then integrated anywhere where there's a double-stranded break made and integrated into the genome. So you can essentially then use that orange double-stranded tag as like a landing pad, and you're able to sequence off of that. And so what IDT has done to improve the technology is highlighted here, and I won't go into a ton of detail. We have a preprint on bioRxiv in the bottom right here. But essentially, we've been able to really improve the sensitivity of this by incorporating things like a blocking oligo to reduce any non-usable reads from adapter tag, by making improvements to the bioinformatics algorithm. And really what this does, it allows us to get about 10-fold more off-target sites nominated than with standard technologies. So this is the nomination assay we used for this child, alongside the IGI and other partners doing in silico and in vitro assays. And so this is actually the data that we provided that went into the manuscript, went into the IND discussions of nomination of this guide RNA using UncoverSeq. this guide RNA targeting this CPS1. And so what's highlighted here is that we did this with the wild type CPS1 guide RNA as well as this patient mutant version. We did this in multiple cell types. And then alongside the other orthogonal assays, we came up with a list of 300 sites based on pathogenic risk, based on where these are these encoding regions. This is the list we're going to do off-target confirmation with on the clinically relevant batches for this patient. And so how do we do off-target confirmation at IDT? We use our RAMP-seq technology. And again, this is a technology that was developed about 10 years ago at IDT. And what this is it's a multiplex sequencing assay and standard PCR library prep for next generation sequencing. But the PCR reaction in the first round of PCR has an RNase-H2 enzyme in it. And what that does is we have an RNA base in our primer and a blocking group, so that RNase-H2 enzyme will recognize that RNA base when it creates a DNA-RNA heteroduplex, and it will cleave that, releasing the blocking group. So what this does, it allows you to do multiplex sequencing in a very high level. So you're able to do library prep on hundreds to thousands of sites in one reaction. So what this does for off-target for CRISPR is it allows you to look at that on-target site, see the editing that's happening there, but then it also allows you to look at these 300 nominated sites that we said these are the sites we need to look at for this particular edit that we're making for this patient allele in all in one library prep reaction. And so this is again published and the details highlighted here. But then this is the data of IDT doing our RAMP-seq service, again, on these clinically relevant batches for this patient. So what's shown here is primary hepatocyte cells of male individuals of a few weeks of age, 6 months, and then I believe about a year. And so what we're looking at the top is the base editing SNP compared to a treatment and a control, and then at the bottom, any indels. And we're looking at those 300 nominated sites to see if there's any significant editing outside of the control that is anything of risk. And we detect nothing in the indels, nothing in coding regions. And up top, there's just a low on-target bystander. So all in all, to say this was very, very specific target here. Okay, and then lastly, this is a nice figure also in the New England Journal of Medicine. How does the patient's ammonia levels and protein intake doing after the treatment? And so in the top left, we're showing the protein intake based on days of life. And so you can see, under the amount of intake that we would like the child to be getting. But following dose 1, you see that, now be able to intake a normal protein level. And again, by dose 2. And what this really amounts to is the patient's body weight starting at the 9th percentile and then prior to the first dose when he was on protein medication and having a reduced protein diet. But then now with the 26th percentile in a healthy baby, and there's chubby cheeks, and it's just very, very rewarding and impactful to see this. And then the ammonia levels following the treatment of this base editor with this guide RNA, you can see after dose 1 and dose 2 in 2 different forms of graphs here, much reduced from the early onset when this was discovered. So this is really a new era for CRISPR, and this is truly a way to make CRISPR cures for rare diseases. And this was one patient, though. This was a therapy developed for him. And how do we take this to many? So as I mentioned at the beginning, IDT, Danaher have partnered with the IGI. How do we platformize this? How can we take what we've learned here and allow this to be much more broadly applicable? So one thing to think about is, could future urea cycle disorder patients that would have a different mutation potentially use the same base editor and LNP, because those are not changing, and we just go and we do the customized guide RNA that has the unique mutation through the validation, through the regulatory de-risking. Could that be an approach? One thing that was announced this year also is there's been a $20 million initiative as part of the Chan Zuckerberg Institute and IGI to really develop these personalized medicines. So this is stuff people are really focused on. And IDT and Aldebaran and Danaher and Cytiva will be there right along the side to produce these clinically grade components. And, kind of like bring it all back together of, again, when I did that first CRISPR experiment, I never could have imagined, like 10 years later, this was going into a patient. We've thought about the right things to be able to manufacture this in the GMP needed setting, to get the right relationships and partners with people in place. Building the right relationships and trust, so paramount as a scientist, working with your community, and then really having these off-target assays really refined as a service and ready to go. Like, the timing was just so amazing. So I want to end on just highlighting here that the single successes are really the beginning of every breakthrough. So on the left, we have Emily Whitehead, who in 2012 was the first child to be cured of relapsed leukemia using a CAR T cell therapy, really transforming cancer care and launching a new era of immunotherapy. And now there's 8,000 to 10,000 eligible patients per year. In the middle, Victoria Gray, she actually presented was present at a meeting in Phoenix, meeting on the Mesa a few weeks ago, all talking about CRISPR therapies, which was just amazing. But she was the first person to be cured of sickle cell disease using the ex vivo gene editing therapy that I talked about at the beginning, Casgevy, which now there's 16,000 eligible patients per year. And then baby KJ. In 2024, he was the first to receive this personalized CRISPR medicine. There he is, looking incredibly healthy, demonstrating that we can do this rapid, custom genomic medicine. And so, obviously, this is one patient. This was made for him. How do we scale that and be able to make these personalized N=1 medicines for many, many children with these rare metabolic diseases? So this is actually an acknowledgement slide from the powerful Rebecca Arns-Nicholas, who presented at ASGCT all of this information in May. And it just highlights everyone, the large group of people that were part of this. Her team at the CHOP section of metabolism, Dr. Kiran Musunuro, who led the team at UPenn that studied this base editor and this therapy. All of the leaders on the top up there that were really the foundational people driving this work, and then the partners at Acuitas, Aldebaran, IDT that made manufacturing this product, doing these services all possible in this timeline. So, bold ideas, brave experiments, endless potential. Like, as I said at the beginning, like, you all have impressed me, and science doesn't wait, and neither do you, so I just encourage you to keep pushing the boundaries, because what you think is not possible really, really can be. So, thank you.